in here. Oh, this is a pull down. Oh, wow. I haven't seen one of these in years. I'm a little gun shy because back in my high school days, I did that one day and the whole thing fell down on me. It came right off, so, come on. Hey there. All right, you survived the exam one. I had to add a lot more points than I wanted to. But as I said in the announcement, there were a couple of factors there. We missed a day of lecture, and they were test bank questions. So although I selected them, I thought they were pretty decent, and then I thought the focus study outline was pretty directive, but it is what it is, and now there's a good distribution of grades. We're not going to get them back today.
Or is it because I skipped the testing?
Welcome. Boy, this has been a sort of a musical chairs today. Thank you. So somebody finally got you, huh?
Yeah. You're wondering where everybody else was. I always have them right here.
Did you curve the exam grade? I'm not sure what you're referencing to about the adding points.
I added 18 points to everybody's score, so the score that's posted has the 18 points.
Okay.
Oh,
I missed
that is
that.
50 points.
It was just as a descriptor.
Good,
Showed up.
good time.
In the email, or is that a Blackboard code?
That was in the posting for the grade, for
Okay.
the exam grades. It was in that descriptor. Was it there? Anybody else see it?
No,
Did you know that I raised the scores 18 points?
I didn't know
That makes much more sense
Yeah.
now that I have my grade. That makes more 18.
Okay, welcome. Sorry about, I guess we got flooded out. I don't know, I guess it's the rain. I don't know what the situation was, but in our regular room, it was flooded. So I was first told 160, and then I guess Dr. Mathis somehow or other ended up in 160. Or did she?
No.
No.
Sign next to it.
There's a sign that says they're going 160, but they went in the class.
There was no one trying to give him money.
I don't
Okay,
think that was
anyway,
next.
we're here. We're gonna go without the projector today, because I wanted to use the dock stand, and I'm not sure exactly how that works in here. And I was gonna do mostly board work anyway. Sometimes I think that actually goes better. The PowerPoints are online for you now for this whole section. I am marking this section now as diversity 1. But this is biodiversity 1. And it's all of the lower stuff in the taxonomy scheme. I'm not saying they're not complicated and they're not important. I'm saying that we're doing prokaryotes today, which are bacteria. And those of you who have me for lab, we also were working with them today in lab. On Friday, we do protists. So it's the kingdom Protista, the eukaryotes. And then we'll be going into the lower, I think it's the lower plants, I believe, from here. Let me look at my schedule and make sure we're right here. Yes, the lower plant diversity, how plants colonize the land. Who's doing 29 for the outline? Which is plant diversity 1, how plants colonized.
I'm doing 1 or 2, I can't remember.
Okay, and somebody doing 2? Okay, so it's one of whichever. Okay, that's good. And then we're doing fungi. And then we're going back to 35, vascular plants. Who's doing that one? Anybody know? 35 vascular plants. And that's more of anatomy of that. And then we have an exam on 225. It also happens to be a Monday. Okay, today, bacteria and archaea. You know the domains that we learned about? There are 3 domains. I'd also put in on that announcement or something that I wrote on Blackboard that I think this portion of material will be a little more factual. You know, evolution gets a little conceptual, ecology can be a little conceptual. For those of you who are more comfortable with fact stuff, I think this is going to be a little bit different here. So these are the 3 domains, and you know, there are 2 different cell types of all living things, and prokaryotes and eukaryotes. EU means true nut. It's like a nut in the middle of the cell, which is the nucleus. Has a true nucleus and double membrane bound organelles. Prokaryotes have no nucleus, and actually no nuclear membrane, which makes really no nucleus, but the DNA is in an area called the nucleoid. That's Nucleoid. Prokaryotic cells do have ribosomes. We also have cell walls. We're going to learn more about the specifics of the anatomy and a little physiology about prokaryotic cells. Eukaryotic, we have kingdom Protista, kingdom fungi, kingdom plantae, and then we have many, many phylums, of course, that we'll be talking about in the animal kingdom. So there are lower plants and higher plants we're going to be talking about. We're going to be talking briefly about the kingdom fungi, and we're going to be talking about the kingdom Protista, like I said, on Friday. So today is primarily focusing on the prokaryotes. And so if I were to ask you, how many domains include the prokaryotes? 2. And what is the name for the 2 domains the prokaryotes are in? Bacteria and archaea. This is kind of interesting because sort of like archaeology, old, but since we have the evolutionary evidence and the DNA sequencing and the comparisons of cell walls, they actually have a different cell wall than, for one thing, than the typical bacteria have. We actually have found that in terms of ancestors, that these 2 groups are more closely related to some extent than the bacteria. But of course, they're both prokaryotic cell types. So in that way, bacteria and archaea are both prokaryotic. but the archaea have some characteristics that are more closely related to eukaryotes. Right, so we kind of used to think in evolutionary terms it was archaea and bacteria, but now it's kind of like bacteria first. In fact, life originated about 3.5 billion years ago with bacteria. And so with the heterotrophic bacteria. And so now we're going to see references to evolutionary sequences, so don't forget all that evolutionary stuff you've learned. I think it's going to make more sense now. So we did this in my lab today, but and by the way, I think we also learned before that the primary reproduction process of bacteria is binary fission. And the thing that you need to know about this is the fact that they have one circular, although really twisted. It's not like a clean circle, it's all twisty, but it is one joined piece of DNA is the main chromosome of the bacteria. And besides that, they also can have plasmids we're going this way now. So in addition to one circular chromosome, I'm primarily talking about bacteria. We're going to spend much more time in bacteria domain than we are archaea. The big thing here with archaea is that they often live in extreme, well actually they're called extremophiles. Some live in heat dense in the earth, and those are called thermophiles. Some live in extreme salt concentrations up to 35% salt, which would desiccate most cells, including ours, since we have 0.9% physiological saline. And those are halophiles. They also have acidophiles. The extreme bacteria that can live in extreme acid. I think the introduction of this textbook in this chapter actually gave you an example of one that was, 0.3, yes, 0.3%. Thank you, Nick, for the outline. It's a good outline. And Picrophilus optionii. How many of you have this already printed off? Good? Good. Okay, so you want to be sure to do that, and there are actually two on there. The next chapter is also on there to copy off as well. Okay, these plasmids are extrachromosomal pieces of circular DNA that a bacterium may have. It's not necessarily always true, but the most famous bacterium of all from genetic engineering standpoints and from knowing so much about it is Escherichia coli or E. coli, genus E. And where does it live? In the colon, the intestinal tract, the large intestine of mammals. And so it's used as a indicator for whether or not there could be fecal matter in water supply, food supply, and we've, in keeping with the news of the last month, 2 months, 3 months, we've had a lot of E. coli outbreaks from lettuce to Chipotle has also been in trouble several times for, and it's not always E. coli, but other types of food poisoning. And what does E. coli look spread on an EMB agar plate?
Metallic green.
Okay, and that's what you're going to say too, right? It's a really pretty metallic green. Everybody seen that at this point? So that's a good detection. It's a gram-negative rod. Oh, okay. Now, ooh, still talking about things. In terms of bacteria, really, the bacteria is divided into 2 big groups. One is gram-positives, and 2 gram-negatives. And gram-positives have a thick there are some good illustrations in your textbook. Be sure to look at the illustrations and figures because they show you the differences. They're also in the PowerPoints. Sort of a thick, you know, whereas the cell walls of plants are made of cellulose, and the cell walls of fungi are made of chitin, C-H-I-T-I-N. The cell walls of bacteria are typically made of peptidoglycan, particularly if they're gram-positives. Gram-negatives have less of this. It's more thin, but sandwiched with other complex peptidoglycan type. It's actually more complex. And as a result, it takes a different antibiotic to attack gram-positives. All you have to really do is to attack the peptidoglycan later. And it's also something we don't have in our bodies, right? Except for other bacteria. Our cells do not have peptidoglycan in them. So when you're designing an antibiotic or designing a drug, either you want to attack a structure that you as a human don't have, so it's not going to be that toxic to you. So you target the cell wall. It takes a different class of antibiotics to go after those as it does gram-negatives. Of course, then we can run into resistance in either one of them. You also many times end up with endotoxins, sometimes released from the cell walls of gram-negatives once they're killed. So you can actually have like a fever that develops after they've been killed because of the homeostatic regulation in the body of the body temperature. Okay, so,
So basically, does that mean that when those bacteria are killed, they release toxins?
right, they do. And there are also exotoxins that other, like some of the gram-positives and some of the others can also release, which just means they secrete toxins into the environment. And a toxin is a poison, so, you know, the people react to. So, for the gram stain, what's the first stain that goes on after you make your smear? Crystal violet, okay. And this is called the PS, or primary stain. It's blue, but it's violet. It's also called Huffer's crystal violet. And then we put iodine on it, Graham's iodine. And this acts as a mordant to further kill, if it hasn't already been killed by heat fixing, but to further kill the bacteria and also to help it stick to the slide, the stain. And then alcohol is the colorizer. If it's, the color will go out if it's a gram negative or positive. Negative, if it's gram negative, this crystal violet will wash out with this alcohol. And then you put a counter stain on, which is safranin.
Is that what gives it the pink color?
Right, so gram positives end up retaining which color at the end? Blue, purple. Our gram positives at the end, and gram negatives will end up with the pink, red. Now, for the lecture exam, I would not ask you how long are we supposed to put the crystal violet on for, or how long. I could ask you what the order is, if we need an order, to be exact. But in lab, you should know how much time we were supposed to leave it on for. Okay, so, questions so far? Yes?
Why does the EMB auger not take the blue color of the methylene blue that's in it? I noticed when we were working with it in lab.
The eosine and the methylene blue, which is what the EMB is. I think it also, it has to do, it's a chemical reaction, but I think it may have something to do with the pH. I'm not gonna swear to that. I'm not gonna ask you that
Because it
question.
usually looks more orangish than it does blue.
Yeah, or purple. But it's a selective stain to prevent gram negatives from growing. Plus it's differential because it allows the green sheen for the E. coli test to indicate that it is there. All right, now this big one I started, I'm sorry, let me do this now. Shapes and arrangements. This is really, really important. Simple morphology. Morphology is the way something looks. This is microscopically. Not to be confused with colonies, which are visible masses of bacteria that have grown for a couple days from a single cell. By the way, you do know, we should probably mention that too, a colony forming unit. Here is, you've all done your ubiquity plates, right? Or something with the microbiology. If it's actually a true colony, bacterial colony, it's like an island. It has nutrient medium all the way around it. It's isolated. That particular colony is considered to be a pure culture. Because if it doesn't have other stuff running into it, the hypothesis is that one bacterium was laid down there, and over that 48-hour period, it divided by binary fission, which could be every 20 minutes, and it produces visible mass. So we know bacteria are, this is something else we probably should say, measured in micrometers, whereas millimeters are 10 to the negative 3. This is meters, 10 to the negative 6 meters. So it's 1,000 times smaller than a millimeter. Or we should say there are 1,000 microliters in a millimeter. Let's say it that way. And bacteria are actually measured, which is 100 times the 10 of the oculars to give you 1,000 total magnification to be able to see these. Now, it might have been possible that back in 1081, that when you did your buccal smears with the scraping of your cheek, and I know the highest we looked at was 400 magnification, it would have been possible for you to have seen little tiny dots in the background, and they might have been in different shapes and arrangements, but you had to really stare hard, and those might have been bacteria that came out of your mouth. Now, if you have bacteria that come out of your mouth, does that mean that you're sick? No. If you have bacteria that came out of with the Ubiquity and Microorganism Lab that came from your nose, does that mean you're sick? No. Thanks to the immune system. No, it doesn't necessarily mean you're sick. If you, with your finger touches, if you did the finger touches, just about everybody, in fact, we've got pores in our fingers, and there's bacteria cling almost like caverns, like caves. They cling to the inside of the pores like they're in caves. And there's some pretty neat electron micrographs that show that. They aren't necessarily pathogenic, which means they cause disease. They may be some other symbiotic relationship. Remember, we just studied symbiosis, where they could be mutualistic in many cases. They could be commensal. They could be actual pathogens, which mean they make you sick. But the immune system tries to fight them off unless they get too many of them, that the immune system can't handle it. So E. coli in the intestinal tract is supposed to be there. It helps us to break down the food so it can be absorbed. And it helps to produce vitamin K, which is for clotting your blood. And it helps in other reasons too. It also helps to cohabitate the surface area of the lining of the colon, so there's not space for other opportunists to get in and colonize. But when we take antibiotics, we also kill a lot of what we call the good flora in our colon as well, and we leave spaces open, and that's where yeast infections and things of that sort can come about, because there's probably always some there, and if there's a lot of empty space now because they've slid off and gone on through the feces, that you've got this empty space, that the yeast can now start budding and they can start reproducing, and yeast infections can result. We're really going to do it this time, okay? 3 shapes. We're gonna use the preferred terminology. We're matrix biology. We don't need to say rod. We don't need to say sphere or circles. We don't, we need to have the correct terminology. And so let's say coccus, bacillus, spiral or spirilla. Can be either one. It just means it's kind of curved in some way. Coccus are round spheres. Bacillus are rod-shaped, more rectangular, but not sharp edges, rounded edges. And spiral can be anything from a comma to an actual spiral shape. Now, bacteria have genes in that circular chromosome. So they have one circular chromosome, true chromosome, and they have genes that actually code for the way they prefer, according to who they are, how they prefer to arrange themselves once they divide by binary fission. So coccus can either be single, what we would call that? A packet of two, diplo, very good. They can be more than two together, a chain, strut. They can be a cluster, like a cluster of grapes, and that's called what? Staff, yes, staff. Now, it's not STAFF, it's STAPH. So you hear somebody's got a staph infection. I wonder what arrangement the bacteria is probably in.
The grape arrangement.
Yes. And if you put the arrangement first and then the formal name for the shape, you have things like Streptococcus, Staphylococcus. So on a test, if I asked you to draw a Staphylococcus, could you do that? You draw a bunch of circles like grapes and a cluster. If I asked you to draw a Streptococcus, you're going to draw coccus around, so you're going to draw something like this. Okay, with bacillus, because of the shape of the bacillus, there's no staff. Okay, so let's just put this no staff. You can have, there are some other arrangements that you can get into, but these are the major ones. Single, diplo, strep. So, Streptobacillus, can you draw that? So it would be okay. Now, I don't want you to get overly confident that this is all this simple. When you, I don't know, if you're in the other lab, you may have already seen your slides. We didn't get that far because we missed last Wednesday, so we're kind of behind. But we've made our grandstand, and we need to look at the slides now. And so if it's a streptobacillus, no, not really. You can find some singles probably. You may even find some diplo. But the question is, when you see a mixture, if it's assuming it's a pure culture, it's the same bacteria. If you see a mixture of arrangements, you kind of stand back from it and you say, which arrangement seems to be the most predominant? Which one's most frequently in occurrence? And that's the one you typically pick to describe and to say it's mostly strepto or whatever happens to be, but some single and diplo present. And that's the way you can do that. Again, this can be spiral, can be comma, or it can be more, it's not a very good spiral. They're all single, by the way. As far as I know, I don't think So those are the shapes and the arrangements. We've got a little bit of material up here. Anything difficult? Well, there could be a lot of questions coming from that, right? What I would like for you to do also is, when you study, is to take the information and try to form test questions. If you're in my mind, I intend, okay, I am going to make up this next test myself. But I'm going to probably throw in a half dozen or so from the test bank. But the majority of the test is going to be my own wording and my own written questions. Probably may be a few more short answers though, and some other types. There'll be some multiple choice. I'll let you know more when we get there. But I plan for it to be different. What do the cell walls do for the bacteria? According to the outline, they maintain shape, which one of the three major shapes. They protect the cell. They prevent lysis, which is bursting, due to osmotic situations. And Nick is going ahead and told you gram-positives have a thick layer of peptidoglycan. The gram-negatives have thin peptidoglycan, and the lipopolysaccharides, LPS, that's also in there too. It makes it more complex. Okay, we are to, let me discuss this since it's up here. These extra chromosomal pieces that can be found in a bacterium called plasmids. There's a portion on the outline where it talks about some of those plasmids can be for fertility. And it's not something that we normally think of bacteria worrying about whether or not they're fertile. Sounds kind of strange, okay? Just hang on. But and some of those plasmids are our plasmids for resistance to help fight bacteria so that they can survive and reproduce. What kind of concept is that? Evolutionary. Survive and reproduce. All right. And also the plasmids are the workhorses of genetic engineering. For instance, and some of you know people who take insulin. Okay. Most likely, the insulin is genetically engineered insulin. And if you have access to one of those bottles, you might want to look at it and see what kind of DNA they say it is. Okay. So what researchers do is they can take a plasmid from a bacterium and they can use restrictive enzymes, which you remember from 1081, probably in the lab, and they cut at certain sequences. You're talking about G's, C's, T's, A's, the certain sequences, and they leave sticky ends. And then you can have the gene for human insulin ready, and you can insert that human insulin gene in, and that plasmid will actually reseal itself, assuming we've got the right enzymes, of course. The ligases and so forth. To put it into the plasmid ring. Every time the cell divides, like every 20 minutes, that plasmid's going to divide. And divide, and divide, and divide, and divide. And you're going to have a cloning situation, and the actual gene product of that gene will be the production of insulin. So it's a protein substance. It's a hormone. And then that can be harvested. Okay, so plasmids are really important to us. And there are actually, and this kind of goes in this area, Ways in which DNA can be transferred. So, taking notes, I would put heading, ways in which genetic material can be transferred in bacteria. And let me take the bottom one first. Conjugation. Remember fertility? Conjugation is a type of sexual reproduction that can occur between some bacteria. And we have this long tubular thing, the pilus, which can actually have genes transferred from a donor, which is a plus, an F plus fertility, to a recipient, which did not have that particular gene before. So there can be some exchange, some sexual, that's a small sexual type of exchange of DNA in some bacteria. Transformation, what we found out is in prokaryotes, there were a lot of historical experiments that demonstrate this. I think it was Griffith, maybe, where if you kill bacteria by heat killing it, and then you mixed it with virulent bacteria, which causes disease, that's very strong, virulent, and causes disease, that when you mixed a live virulent with a dead non-virulent, the dead non-virulent actually absorbed the genetic makeup of the live ones, and they were transformed. And so prokaryotes have that ability to actually absorb DNA from other sources. And that opens up a whole new world. Okay. And transduction usually has to do with viruses. Remember, viruses are obligate intracellular parasites. We don't call them living, but they attach to the outside of a host cell, and they inject absorbed into the bacterial cell. That's called transduction. So those are 3 ways of doing that. Also, horizontal gene transfer can occur. I just told you that non-pathogenic bacteria can turn into pathogenic by absorbing. So that's horizontal gene transfer too. And remind me, if you would, to talk about the positive and the negative importance of bacteria before we go to Protus on Friday. We don't do this Friday, I don't think, because we have an exam Monday. But keep studying, don't get behind, because it's still like a chapter. Thank you very much. We will see you.